And somebody said they asked what color notebook.
Blue.
Hey, Dr. Roberts, can you hear me?
Yes, we can hear you.
Okay, just making sure.
Yeah,
There's a
good thing to check.
well, it would have been okay because I would have been okay with it.
Well, you know, you are getting part of your grade from it, so you probably want to be heard.
I mean, eventually I would have figured it out, or somebody would have been like, "He's not talking."
Okay. Mert, I'll turn the controls over to you, but Jonathan will be our first speaker today.
Sounds good.
All right, thanks.
All right. Thank you, Ty. All right, so today I'm going to talk to you about the Deepwater Horizon oil spill and how it has caused accelerated erosion rates in saltwater marshes in coastal Louisiana. So, Mert, can you go ahead and go to the next slide for me? Mert. Oh, okay, good. Sorry. So to begin, I'm going to go through an outline for how this presentation is going to unfold. To start, I'm going to go over some basics of just what happened with Deepwater Horizon, when it happened, where the area of was affected. And then after that, I'm going to move into how to classify oils, sweet versus sour, in the class system that's used. And then moving forward, we're going to go into the effects that Deepwater Horizon had on the saltwater marsh environments, in particular the vegetational impact and also land loss impact. And then after that, we'll wrap it up with conclusions and references. Mert, can you go ahead and go to the next slide for me? So for basics of the spill, the day that it occurred was on April 20th, 2010. And upon the mobile drilling unit exploding and catching fire and sinking, it caused the Macondo oil well to actually leak freely into the Gulf. Not just oil, but other substances as well. And efforts were made to cap this well, but were unsuccessful. And this allowed the well to actually leak for 87 straight days. So the well is located about 50 miles off the coast of Louisiana. And if you turn your attention to the image on the lower right side, this is just a rough estimate of like showing where the oil has spread to. Okay. And it was estimated that 3.19 million barrels of oil were spilled in the entirety of this endeavor. Mert, can you go to the next one for me? I'm sitting here trying to click, and I'm like, you don't have control. So when looking at classifying different oils, you can look at sweet oil versus sour oil, and then there's a class system that goes from A to D. So looking at sweet versus sour, when you think of a sweet oil, it has a lower concentration of sulfur versus a sour oil, which has higher amounts of sulfur. When you when they're classified from class A to class D, class A oils are usually light and they exhibit a very liquid behavior, and that allows them to move very quickly across surfaces of water like ocean, and it allows them to spread faster. They're actually very volatile and flammable, and this allows them to be, this causes like a very bad like danger. Oh my goodness, I had that worded so much better in my head. They actually pose very high threats, I guess is how I want to say it. And as you move from class A to class D, the oils exhibit more of, instead of a light weight characteristic, they become more heavy, and their physical characteristics change from being liquidy to becoming more waxy and oily or thick, sorry. And when you move towards from A to D, the ability for it to be absorbed into like a porous surface, such as the sediment, it becomes a lot harder, and temperature kind of becomes a factor. So Mert, can you go to the next one? Okay, so this is just to segue into our next area, and we're going to talk about first vegetational impact, and then I'm going to move into a study of Spartina alterniflora, and then I'm going to go into the resulting land loss. Mert, you can go ahead and go to the next slide for me. Okay, so here you can see a couple of different vegetation sources that were affected by the oil spill. In the top left, you can see the Spartina alterniflora, and you can see that it's actually located right there on the bank of the marsh, which is going to become important in this next slide. And you're going to have to forgive me, I'm probably not going to say these names right, but I'm going to do my best. In the upper right, you can see, I believe it's Juncus ramecnerus, or I don't even know how to say that. And you can find those in brackish marsh. And then on the bottom left, you can see Phragmites australis, and that's intermediate marsh. And on the bottom right, I think it's Avicenna germanus, and that is found in saltwater marshes. So, Mart, if you can go to the next slide for me. Okay. So with this, I want to start by looking at the changes in the chemical components of Spartina alterniflora. Then I want to move into an analysis of how Spartina alterniflora was lost due to the oil coverage. And then I want to finish up with the role that Spartina alterniflora play in soil stability. So I found a study of Spartina alterniflora, and it was done by Brittany McCall and Stephen Pennings at the University of Houston. And their findings were that soil organic content, water content, and soil salinity did not differ significantly from before the spill versus after the spill. There was a slight change, but in terms of statistical analysis, it wasn't outside of the range of significance. What did change, though, was the leaf nitrogen content found in Spartina alterniflora. And the reason I wanted to bring this up was just to show you that there was a chemical change to this vegetation after the oil spill happened, even though there wasn't a very significant amount of change to the soil organic content, the water content, and the soil salinity. So now if you could bring your attention to the image on the lower right side, the three separate images that are side by side, they go hand in hand with the schematic that's above that. So just bear with me and I'll get to the schematic in a second. So also in this study, they analyzed the loss of the Spartina alterniflora because of oil coverage. On the far left, you can see healthy Spartina alterniflora that actually that didn't come in direct contact with the oil. In the next middle photo, you can see where the Spartina are actually kind of coated in the oil. So you can see those dark spots closer to the bottom. And then on the right, the very far right, you can see where the Spartina alterniflora have actually not only died, but they've actually they're just gone. They've been like washed away, and that's not a good thing for soil stability. So in the corresponding picture above, you can see the schematic of how the healthy Spartina alterniflora are far, far away from the water edge. And then you can see that region in the middle where there was oiling, but they're still partially there. And then there's that zone on the right side where there's no Spartina left. The dimensions of these zones are from water edge to the oil Spartina that are still there is 5 meters wide. Oiled Spartina that are still there are 2 meters wide, and then healthy vegetation made up the remaining land mass. Okay, so now if you could look at the left image, you can see above and below ground biomass for Spartina alterniflora. And what this role is you can see in the below ground biomass, there's a very vast system of roots and rhizomes. And what this does is it helps hold all the different components of that marsh soil together. And it actually acts as a deterrent for erosion. So it helps to prevent heavy erosion from taking place. So you can see how, since there's no Spartina left on this marsh edge, how there's going to be sort of a lack in that soil stability, which is going to make it more susceptible to erosion via tidal waves, like as the tide changes and as waves hit the bank. I also want to talk about the above ground biomass. I found that in this study, they found that as the tide goes up and down and brings soil into it, the stems can act as a way to trap soil from going back out. And what what I found significant about this is, as the sea level rises, this is going to actually help the marsh sustain its structure as the sea level goes up, because it traps the soil and causes the marsh to actually retain more sediment and soil. So, Mert, if you could do me a favor and go to the next one. All right, so this is going to be the resulting effect on the Louisiana saltwater marshes. So to start, I want to go over the mechanism of how the shoreline is going to erode after it's been introduced to oil. And then after that, I'm going to talk about how the oil and the soil actually interact. And I'm sorry, I'm having a dry throat problem. How oil and soil actually interact. And then after that, a comparison of high oil sites versus low oil sites. So if you bring your attention to the bottom right side of the screen, there's a schematic, and we're going to start with the top. And you can see that there's no oil being introduced. That's that smooth line. And there's no oil being introduced. As you move clockwise to the right, those dashed lines represent where oil has been introduced into the marsh bank. So yes, good job, Mert. On that image to the left, that's exactly what it looks like, those dotted lines. And that's actually going to be the site where it's going to break down first and erode away, because since, like in the previous picture, that where Spartana had completely washed away or was not present, that's where erosion is going to actually start first, since the vast network of roots and rhizomes are not there to hold it together. So if you move to the third image on down, keeping with clockwise motion, there's going to be down right. There you go, Mert. There's going to, after those two pieces erode away, there's going to be a non-oiled head. And what's going to happen is, this is going to be the site where a concentrated wave is going to hit that first, and it's going to cause it to actually erode away at a very quick rate. And that's the non-oiled heads you can see in the image to the left, is basically the green spots that are still even with the water. In the fourth image, or the fourth part to the schematic, you can see a complete breakdown of that non-oiled head. Yes, right there. And that's actually this big gap now. And as you move up to the next one, yeah, right there, you can see that that dotted line has actually moved to where all of it was actually non-oiled. And you can see where it's had this cascading motion, or motion, and it's broken down where it previously was, and it's moved back. And then it'll eventually come to an equilibrium when erosion will occur at almost the same rate all the way across, at an even rate. So now I want to move into how when oil is introduced to the bank, how it interacts with the soil. So when oil washes up, it actually brings with it this it's not abundance, but extra nutrients. So instead of roots being produced going further down into the soil, since there's these extra nutrients that are on top, it causes more of a top-heavy biomass. And the problem with that is, if roots aren't growing straight down and they're not continuously supporting the soil, it can allow it to erode away from underneath. And what you would see with that is an overhang. So after that, I want to move into a comparison of high oil sites and low oil sites. I found a study that was done via LSU, the Department of Oceanography and Coastal Sciences. And their study was from 2010 in November to August 2012. And what they looked at was sites where low concentrations of oil were found versus high oil concentrations. And in their study, they actually found that in the first 3 months of the study in its entirety, sites that had low oil concentration actually had a higher rate of erosion versus that of high oil sites. But in the last 4 months, sites that possessed high oil concentrations actually exhibited a higher erosion rate. But for the study in its entirety, sites with low oil concentration had the lower rate of erosion versus sites with high oil concentration. And those rates are 100 centimeters per year at low oil sites versus 133 centimeters per year at high oil sites. And yeah. Okay, that's all I wanted to say about that. Mert, we can go to the next one. So to wrap things up, I feel like I'm a little bit over on my time, but to wrap things up, we talked about a few different vegetations that were affected by the oil spill, and we moved into a more specific analysis of Spartina alterniflora and how they actually play a really good support for soil stability along the marsh edge. And with the loss of that, we moved into shoreline erosion and how it can be done at an accelerated effect after the Spartina has actually been removed from the marsh bank. And Mert, that's all I have. So I'll take any questions.
Hey, Jonathan.
Yes, sir.
So you mentioned 4 species at the beginning, which are obviously just a subset of the species that exist out there.
Yes, sir.
In your research, what did you find about the relative susceptibility of them or sensitivity of those species to oil? Did you get into that at all?
I actually did not go into any susceptibility with that, with other types of vegetation. The reason, there were, as you know, a ton of different vegetations that were actually affected by this, but I didn't have a lot of time to go over that. But with the Spartina alterniflora, I actually didn't go into it as much. I found that what happened with their high mortality rates, it was due to being covered and photosynthetic processes being inhibited, which actually caused them to die off at a faster rate. But if I had more time, I would go into more of a susceptibility to that oil.
Okay. Let me just say, just for general knowledge, that there is a very large gradient in susceptibility to oil. And the species that you had there, you had Phragmites australis and Spartina alterniflora are 2 of the least sensitive to oiling. It's part of the reason why Spartina in the bioremediation can be successful. You also had Juncus vermerianus, which is a highly sensitive species, but perhaps the most sensitive species that you have there is the black mangrove. Mangroves are considered to be more sensitive and have longer recovery times than different marsh-associated plants, seagrasses, pretty much anything. So just for general knowledge, there's a very large gradient in there, and there's a wealth of literature on this topic with a lot of that research being done by people here in Louisiana.
Yeah,
since we have a lot of oil exposure for a long time.
Yeah. When I was looking through it, it was so much information. I was like very overwhelmed since I was finishing up teaching last week. But I mean, I wish I would have had this topic when I was down there, so I could actually utilize the resources at the Marine Center. Just to have more, I don't know, I guess an abundance of stuff, so I could have asked questions and became more.
Yeah, that's fine. I just wanted, since you presented the 4 species, I want to make sure people at least had some idea about the variation that existed between those. Any other questions for Jonathan? Okay, thank you very much, Mert.
Thanks, Brian.
Okay, so this is my presentation on the challenges of the Pacific salmon populations. I am going to talk about overfishing, which has been a problem for them um, in almost all their areas. Some of the nets, the gill nets in the upper left corner, the fish wheel, basically because these will basically grab them and it will grab a large amount of the salmon as they move upstream. The dip netting, this is a summer pastime in Alaska where a lot of people go out with dip nets, and more salmon probably escape that. The picture on the bottom right is actually a salmon run from Kodiak, Alaska in 1943, which I know it's hard to see the top part of that picture. And then the picture on the bottom left is it basically is showing the Chinook salmon runs in California, and this pink area up on top is completely blocked by the Shasta Dam, and there's one other dam up here that I'm trying to remember that I have forgotten the name of. But a lot of the salmon runs in California are in trouble, and they're in trouble for a lot of reasons. But one of the issues is the damming of the streams and the destruction of the streams. So I'm going to go through some of the populations, their life histories, and the challenges of the different types of salmon. And then I'm going to talk about how different areas have tried to maintain their salmon populations. So if we could go to the next slide. This is the Chinook salmon, which is important all up and down the West Coast, and all the salmon species that I'm showing you they're all around the Pacific, so they're not specific to Alaska or California. Those are just the areas that I'm concentrating on. The next slide, please. The life cycle of the Chinook salmon, most spend 1 year in freshwater with 3 or 4 years in saltwater before returning to the freshwater to spawn. Some females will spend a longer period in the marine habitat and maybe 7 years old at spawning. Some of the graphs over here, the bottom graph shows some of the historic takes of Chinook salmon over time. When you start out, you've got almost 25,000 on this side, and then when you come over here, we're at less than 10,000 units. Basically, it's crashed. Now, a lot of this green part here is being maintained by New Zealand farmed fish, which is the U.S. does not farm Pacific salmon. Ours is actually, well, I didn't find any, but the New Zealand does quite a bit of farmed salmon, and I think there are some Asian countries that also do that. We could advance to the next slide, please. Okay, Chinook salmon runs. This is comparing the Chinook salmon runs in Alaska, which are mostly up the Yukon River, and there are, I'm sure that there are some issues over here, but there are no populations that are in trouble in Alaska. In California, the areas here above this point with this lighter blue, this area is currently blocked. It doesn't really show it here, but also these bottom areas that are blocked. These are historical ranges, and the bottom areas are blocked mostly due to the drying up of the streams, and the upper areas are blocked mostly due to dams. Okay, and we're going to advance to the next slide. Okay, this is another slide that shows historical differences. The green bars, this actually is from Washington State, but I picked it because it was just a really clear representation of the differences. The green bars are all from historic estimates, and the white bars are from the recent runs, and it really gives you an idea of how much these populations have shrunk in a good part of the U.S. Let's advance to the next slide, please. This is the coho salmon. This picture also shows a lot of salmon change as they come upstream. Their color and their beak changes quite a bit, so the color changes as a signal that they're ready for spawning, and the beak is how they defend their spawning grounds. Let's advance to the next slide. Okay, coho salmon spend about 2 years in freshwater, often in lakes. If you remember, the Chinook were only spending about 1 year, and mostly in streams and rivers. The coho are going to spend more time in lakes, and before they migrate out to the saltwater, to the marine environment for about a year. So they're going to have we have 5 different ones, and they all have different times spent in freshwater in different areas, but they're all, in a lot of cases, they're all using the same rivers and streams. There are 4 coho salmon populations on the West Coast that are listed on the Endangered Species Act, and these are the lower Columbia River population, which is threatened, the Oregon coast population, which is threatened, the southern Oregon, and the northern California coast population, and the central California coast population, which is listed as endangered. And I've actually read some things that suggest that it's may or may not actually be able to come back. Next slide, please. Okay, the Central California coho salmon runs. Basically, everywhere where there is a little stop sign is blocked for the salmon. So these are all historic runs, and then these dotted areas are blocked due to drying up. So all of these areas would normally have healthy salmon runs, but the habitat is degraded. Next slide, please. This is the humpback pink or humpback salmon, and they develop this huge, deep-bodied hump as they get close to spawning. The males do anyway. Next slide, please. Pink salmon have an obligate 2-year life cycle, where they basically are spawned and swim out to sea almost immediately, and they return to the saltwater on their return from the saltwater on their 2nd year. Basically, the obligate 2-year life cycle means that these salmon have populations that may spawn in the same streams but are genetically distinct. So protecting, you may have one run in that seems that because they're using the same stream, it seems like these salmon may be actually the same, but because the every other year group, they're actually, it's kind of like the cicadas, where they're different genetically from year to year. So your even year fish are different than your odd year fish. In California, this species is commonly, or was historically commonly seen in Northern California, extreme Northern California, but it is not commonly seen. I think when I looked at 2016, they saw like less than 30 in California. In the Yukon, they spawn close to the ocean, and then obviously go right back out to saltwater. Next slide, please. And then the last one, the sockeye salmon. You see a lot of these because of their bright red color. And this picture gives you a good idea of the differences in how they look. In the ocean phase, they're much more silvery with less of an extreme beak. Next slide, please. The sockeye salmon spend a significant amount of time growing in freshwater lakes, and their dependence on lakes kind of sets them apart. They're going to spend as many as 1 or 2 years in freshwater, and then 2 or 3 years in saltwater, meaning that they're going to come back. In some cases, they're not going to be they're not going to come back till they're 5 or 6 years old. These actually are. There are certain populations in Colorado. There's a population that it was because they can live their entire life in freshwater. They've stocked a population in California. There are a few populations that are natural, even though most of the landlocked salmon did seem to have a lot of hatchery supplementation. And when they're significantly smaller than the saltwater fish. Next slide, please. And the last one, the chum salmon. These are also referred to as dog salmon. Next slide, please. So these also have sort of limited use of freshwater. They move to the ocean really soon after hatching, but they spend 3 or 4 years there and spawn on their 4th or 5th year. This is another one that has been pretty much eliminated in California, and it has some populations also in the Oregon and Washington area that are listed with the Endangered Species Act as threatened. Next slide, please. So some of the challenges facing all the Pacific salmon currently, one of the main ones is habitat degradation. Overharvesting has been a problem basically since the early 1800s or mid 1800s, and as populations dwindle because of other problems, while we're harvesting maybe only a few 100,000 in some cases of certain species, and historic populations, they might have been harvesting in the same area. They might have, in 150 years ago, they were harvesting over 10 million. As the habitat is degraded, overharvesting can mean, harvesting at all can be a problem. The habitat degradation that has been a problem is altering the stream channels. A lot of that has to do with mining. When a lot of the fur trappers, when the beaver populations are reduced because salmon prefer to spawn in streams that are cold and that have significant woody material in them, the destruction of the beaver populations can actually have a significant effect on salmon. The mining operations from the gold rush in both pretty much along the entire West Coast, the gold rush in California in the 1840s, and then the gold rush in Alaska about 45 years later, or 40 years later, all had significant effects on the salmon populations, because when these streams are when they're dug up and a lot of sediment is dumped into them, when wagons or now the heavy equipment is driven through them, it chews up the stream and dumps a lot of silty sediment into it, which it's a salmon habitat in most cases is rocky, gravelly bottoms and cold, clear streams. They really don't like it when there's a lot of silty sediment in it, or when the water gets cloudy, and the insects and the other prey animals that they feed on at these stages also don't like it. So that causes problems for their food sources. Dams for hydroelectric power.
It's good. We appreciate all this going on here, but you're going to have to wrap up here in just a couple of minutes so that we have time to make sure that we can get the other two presentations in.
Okay, thank you.
Okay.
Okay. And let's move on to the next slide. Moving on to the next slide. Okay, so historically it was recognized as a problem. Can you go back? I'm confused as to where you are. Okay, so basically they attempted to start hatcheries in California very early to offset the problems of overfishing and stream destruction, which was noticed as a problem. The stream maintenance was pushed as a civic duty. Basically, that was never really overcome in California. The fishing and mining interests, and eventually the hydroelectric dam construction, those interests went out. The canneries also had basically pushed a lot to keep the limits high. Next slide, please. So it was recognized within 5 years of the gold rush. The first California law was enacted in 1852. And it was called upon all citizens to clear the streams, but it didn't really happen. And next slide, please. Basically, anytime when you had a significant population increase along the West Coast, the exploitation of the salmon and the habitat degradation, and this should say, it shouldn't say drift nets, it should say gill nets, have blocked the salmon from reaching spawning grounds, which basically progressively reduce the populations. Next slide. So problems with hatcheries and artificial rearing of salmon, because hatchery salmon have less genetic diversity, and in some cases they are not pulled from the same population as the fish they're bred to, or so you have salmon that are hybridized from different populations, which causes problems with when they're released. They may not know where to go back to, so that they're wandering is it disrupts their homing ability. Next slide. Okay, so Alaska similarly started fish hatcheries. They have had significantly less disruption of streams, and so their fish populations rebounded from overfishing fairly easily. However, their fish hatcheries mostly try to manage their releases to minimize the interaction of hatchery release populations and wild stocks, even though it does occur. It's considered supplemental. It's intended to reduce pressure and supplement canneries mostly with the hatchery releases, and they've had a lot more success with managing their population, probably mostly because the habitat is less degraded. Next slide, please. Okay, so the problems of dams and hydroelectric power, which is another major thing that blocks the salmon from reaching their spawning grounds. This is the Shasta Dam. Basically, the mitigation features besides the Coleman hatchery were basically never implemented, and that completely blocked approximately a quarter of the salmon streams for Upper California, Northern California. Next slide, please. Okay, last slide, and this is basically showing mining damage of vehicles through the stream, and the sediment coming down into the stream. And if you remember the beginning slides that showed the fish pictures, those are basically an example of the water quality. Those streams were basically glass clear, and in a lot of cases it's glacier melt or snow melt that's coming down from the mountains. So that's the kind of water quality that salmon spawn well in. They don't spawn well in these streams where the sediment is stirred up and the bottoms are muddy. And so that is my presentation on the challenges facing the Pacific salmon. Any questions? Oh, I'm sorry. I forgot. That was a regulation about the Alaska regulations in comparison, which actually the Alaska regulations on fishing salmon are pretty loose compared to a lot of the California regulations. They allow quite a bit of mining, but there are fewer people up there doing it. It's also a much larger area. And in a lot of cases, the public areas, they don't allow large machinery or any mechanics. So you can use a gold pan, and in some cases a small sluice box, but in other places they don't allow even that. Now, obviously, if you've watched Gold Rush on on the television. There are mining operations on private property that are pretty destructive. But this Denali Park slide, that was pretty typical of mining regulations in Alaska on public land.
Okay, well, thank you very much. We really need to move on so we can make sure we can get the other two presentations in.
Hi, can everyone hear me?
Yes.
Okay, awesome. So I am Andrea Santorello. I'm a graduate student at UL Lafayette, and I will be talking about how seabirds are useful indicators of local marine ecosystems. Next. So what makes them a good indicator? They are a higher trophic level species that feed at different trophic levels depending on the bird species and even time of year. They are a very visible animal. Their breeding behavior makes them very easy to observe. They breed in colonies on islands off the coast of continents all over the world. This makes estimating breeding populations and breeding success of these organisms relatively easy. Many breeding populations exhibit some levels of allopatry, which means they will often come back to the same island each year to breed. The breeding season takes up a good portion of the year, often early March to early September. Their foraging behaviors make them particularly good indicators of their local marine ecosystems. Many bird species will actively select for one prey species. This makes them good indicators of that particular species of fish. Often they select for fish. They forage in areas around their breeding colony. Some bird species bring back whole fish that are easy to observe and identify when they bring them back to the nest. For those that regurgitate or hold their prey in a pouch, like pelicans, we can collect feathers or fecal samples on the colony to determine prey composition later in a lab. Many seabirds migrate each year to their breeding grounds, and they are often allopatric to stopover locations along the migration route, which makes them a good indicator species for several different locations each year. Next slide. This is a graphic showing examples of different foraging behaviors or predation strategies and the different trophic levels that each bird family will prey on. Next slide. As I said before, many seabird species are selective predators. They predate a preferred prey species. The preferred prey species changes based on location. These are examples of different breeding colonies all over the world and some of their preferred prey species. It's important to note that one bird species might select for a different prey species depending on where they breed. Now we will go through a few case studies using seabirds as indicators of their local marine ecosystems. Next slide. So the first case study takes place in Machias Seal Island in Maine. On this island, there are many different breeding colonies of seabirds, including the four that this study focused on, which is top left common tern, top right Arctic tern, bottom left Atlantic puffin, and bottom right the razorbill. Next slide. So this study focused on the breeding success of these four bird species as a function of the energy density of herring. This first graph shows that for all four bird species, herring was the preferred prey species. Herring is the one at the bottom left, and most of them for most years, each bar is a different year, herring was the one that was most commonly predated on. This is with the exception of the last 2 years of the study, which was 2000 to 2002, where herring was no longer the preferred prey species. I'll talk more about that on the next slide. The second graph shows energy density of herring at the top and breeding success of the four bird species at the bottom. You can see a parallel pattern between the two. And when I say energy density of herring, I mean the content of the herring, what they're made of. So for bird species, a high lipid content is a higher energy density of herring because it's more energy for that fish that's being brought back to the chick. So if a herring with higher lipid content is being given to a fish, that chick gets more energy out of it than a similar herring with less lipid content in it. Next slide. So this is where I'm going to talk about 2000 to 2002. There are 2 lines for each bird species, and all the lines depict the same thing as the last slide, which is breeding success as a function of energy density of herring. This is through the years. So the solid line shows all of the years of the study, which includes 2000 to 2002. When they took out years 2000 to 2002, you get the dashed line. And the dashed line shows a better relationship, better positive relationship between breeding success of the species and energy density of herring, especially for common and Arctic terns. You can see the R-squared values are very high for an ecology study, which is 0.8. So this is just another example of how you can tell, based on breeding success, just how important their selected prey species is. And the main point I want you to take away is that marine birds that select for certain prey species can be a good indicator of the body condition of that species, which then translates to the health of individuals in that population of the prey species. Next slide. The second case study takes place in the North Sea. Many different marine birds breed on colonies in and around the North Sea, and most of them selectively prey on a species called sand eel, which is also known as sand lance in the US. There was also a large commercial fishery of sand eel in the North Sea during the time of this study. Next slide. This is a table showing the different bird species breeding in the North Sea and their breeding success during years of low and moderate sand eel abundance. So if you look at the 3rd column and the 5th column, those are breeding successes of these different bird species during those times. So the 3rd column shows breeding success from 1986 to 1990, which were years of very low sand eel abundance. I'll tell you why on the next slide. And then the mean breeding success from 1991 to 1997 is when there was a more moderate abundance of sand eel. You can see it's kind of shocking. The first few species had low to no breeding success when there was low abundance of sand eel, and they started to come back with breeding success when the sand eel abundance started to come back as well. So you can see a correlation between these bird species breeding success and sand eel abundance. Next slide. So the reason why sand eel abundance was so low from 1986 to 1990 was because of the commercial sand eel fishery. The left graph shows the sand eel landings from the commercial fishery from 1952 to 2000. The second graph on the top shows the same thing, the landings of the sand eel, and the bottom graph shows the breeding success of one seabird species, the black-legged kittiwake. And you can see that they're inversely correlated. So when the commercial fishery had more landings of sand eel, the breeding success of kittiwakes went down. The point here is that seabird species can be a good indicator of prey species abundance. It also gives us a good idea of how much the commercial fisheries are impacting the population of their catch. If there were still plenty of sand eel to be caught during these years, the breeding success of these seabirds wouldn't have been so low. And because of this particular case study, or this particular event in the North Sea, the sand eel fishery was actually closed in 2005. Next slide. So the last case study discusses the possible benefits of using seabirds as indicators of harmful algal blooms and pollutants. This table, which is also continued on the next slide, shows different bird species and their physical reactions to local harmful algal blooms. Oftentimes it's death or neurological issues, and as well as things like flimsy eggshells and infertility. The idea here is that seabirds can be an early warning sign of harmful algal blooms in a local marine ecosystem. Next slide. And this is just, it's just to emphasize that there's a lot of different bird species in a lot of different areas that can be indicators of harmful algal blooms. Next slide. Because seabirds are higher trophic level species, they exhibit high rates of bioaccumulation. We can use them to be an indication of levels of pollutants in an ecosystem through time, as shown by the first graph, which depicts levels of mercury and Atlantic puffins from 1850 to 1990. They can also be an indication of levels of pollutants geographically, which is shown by the second graph, which depicts levels of different pollutants in oyster catcher eggs along a geographic gradient. Next slide. So to wrap up, seabirds can be indicators of prey population density and body condition, or as others would call it, health. They can be an indication of how impactful local fisheries are on the marine ecosystem that they're fishing in. They can be early indicators of harmful algal blooms and levels of pollutants in their environment. The big takeaway is that it's all context dependent. Different bird species have different preferred prey species based on their location. Next slide. So I'm going to bring this graphic up again. For example, if you look at the common murre on this graphic, they can select for rockfish, sand lance, capeland, walleye pollock, or sand eel, depending on where they breed. This local context can be helpful as well. It allows them to be indicators of local fisheries and local health concerns, as well as local ecosystem health. Things that are changing or will change at a certain location may be better monitored by local breeding bird colonies. For example, here at home, habitat restoration on barrier islands on the coast of Louisiana may have many different unforeseen impacts. Some of these impacts on local marine ecosystems can be easily identified by monitoring local breeding seabirds like the brown pelican or least tern. And that should wrap up my presentation. If anyone has any questions.
Do we have any questions? Okay, well, thank you very much. And we get to see Mert's computer.
Sorry about that, unless you want to do my report for me.
No, it's okay. I got enough of those to do myself.
Come on, Brian. Here we go.
All right, and Damon will wrap us in here.
Hey, hello, I'm Damon Morrison. I'm from the University of New Orleans, and this is a presentation on a couple of invasive primary consumers in Louisiana. Next slide, please. Okay, it's important to consider the role of primary consumers, as they are in the second level of the food chain, as you can see in that graph to the right. They're basically herbivores and omnivores that feed on plants. They're ecologically controlled for plants and food for higher level organisms. And they insist on balancing nutrients at the trophic level. So if we get rid of these guys, then they can have effects on not only the plants that are under them, but also the higher trophic level organisms. Next slide, please. Okay, and I know that Dr. Rabelais went over a lot of negative impacts of invasive species, so I won't go over those. But particularly, I want to emphasize this invasion curve. So normally what happens is we have an introduction of the invasive species. We don't detect them until after some time when we can actually notice them. And at this detection point, this can either be by local fishermen or by scientists. But until public awareness begins and we actually get funding for these things, or start doing the research, by that time eradication is extremely unlikely. And this presents a problem in a lot of areas where finally at the top of the curve, we only have local control and management, which is possible. And basically you're just going in a circle trying to get rid of these things, and you don't know if they're going to be reintroduced, and it costs a lot of money and time, when at the point of detection we probably could have gotten rid of them a lot easier. And especially concerning primary consumers, because usually things like snails and insects are hard to see or hard to get rid of, and we don't really understand everything about them biologically. Next slide, please. And the first species I'm going to talk about is the island apple snail. We also, Dr. Rabelais also talked about this, but I'm going to shed some further light on these. They are native to South America. They're the largest of the ampullarids, so they're over 70 millimeters. And these are the largest snails that are currently in Louisiana. And they possess a gill, lung, and a siphon, which is important because they're amphibious, and they can breathe air through a little siphon that they have. And they come out of the water to lay their eggs, and that's when they make use of their lung. Another important part of it is that they're one of the few snails that we have that actually have an operculum, which is, if you look in the bottom picture, it's a little door that can close, and these organisms can actually bear themselves in the sediment when conditions are unfavorable, so when it gets cold, and they can hibernate for a few months. So even if the entire water body dries out, they can hibernate and then reemerge after and take advantage of the situation. So they're mostly freshwater. However, they start to die in 15 salinities, and they do show a cold tolerance. So these things have been shown to actually select through cold weather and actually become tolerant to colder temperatures. Next slide. So these things, with these particular organisms, a lot of snails are hermaphrodites, or they can clone themselves. These guys actually have a male and a female, which nuptial feeding occurs, in which the male actually offers the female a meal in exchange for mating. And these eggs can get actually pretty big, and you can see a cement structure there with eggs that are all laid outside of the water, and it can contain anywhere from 200 to 4,000 eggs. And anything that basically sticks out of the water that is bordering the water, that is heavy enough to hold the snail while it lays their eggs, their snails will lay their eggs on it. And a lot of times we can have up to 70% hatchling success. So if you think of the amount of snails where in some of the water bodies present, even from that picture right there, there could be several dozen eggs, and if each one of those has a 70% hatchling success, that's a lot of invasive snails that are contributed to the environment. Next slide, please. Okay, and here's some graphs, and we talked about some effects from them, but what we didn't touch on is the concerns for bottom-up effects. So what we're concerned about is that these things have the ability to reduce macrophytes or aquatic plants, and through that reduction, we see an increase in nutrients, and from that we can see an increase in turbidity, alter phytoplankton and zooplankton communities. So in the first section of graphs, you can see where plant cover. Now these snails were placed in compartments in real life. Outside in ecosystems, and you can see where plant cover is reduced by the snails. We have increases in total nitrogen and total phosphorus, and also phytoplankton. However, there are some studies conflicting this, but both authors agree that phytoplankton and zooplankton communities are altered, which can alter the food web. The types of fishes that feed on these things or other organisms. In the second set of graphs is basically all ecosystem modeling. So none of this was done. This was done based off of studies, but none of these studies have actually physically been done. So these are theoretical approaches to what these are doing. And the reason why we have that is because there's a lot of systems where these invade. We do not have data previous to the invasion. So that's to bring home the point that with a lot of these invasions, it's important that that we have data in the beginning, but if we don't, then we have to rely on these studies that may not actually reflect their effects. Next slide, please. Okay, and some controls that people have tried some physical removal as the snails are large, but however the juveniles that hatch are at least a millimeter, so if you don't get rid of those, then you're not going to get rid of the snails. Molluscicides, which pose a problem because it can kill other snails. Copper, and if you look at the very top picture, that is actually an apple snail that is floating to avoid copper that is at the bottom. We also have some natural controls by native organisms, and the bottom two pictures of the birds. The first one is a limpkin, and we've actually just started noticing them in Louisiana, and they are actually eating the snails. And the second picture is a snail kite, which is a native bird in Florida that specifically feeds on apple snails, because there is a native apple snail that is a completely different species than what we're dealing with, but it's limited to Florida. And if you look at the bottom two left pictures, the first one is the middens, and those are always made by mammals, so we believe those are raccoons. And then the second picture is actually predation by a bird, where it's selected against the harmful toxic part in the female that produces the eggs. Next slide. Okay, and the next guy we'll get to is the Roseau cane mealybug. And it's important to remember that native scales are beneficial to the ecosystem. So, and they're very highly diverse. And if you look at those two pictures, you can see we get some pretty strange-looking creatures. They're usually very small. They feed on the saps of plants, and they can be food for other insects or wasps. And some economical uses for them is we use them for dyes, waxes, and shellacs. Next slide, please. Okay, and so the studies on this particular species that is currently invading Louisiana, and especially concerned at the Birdfoot Delta, is that for this species, there's not a whole lot of research. We just know that they're native to China and Japan, and we started noticing die-offs in late 2016, early 2017. And as you can see in the top map, they've spread quite rapidly up through the southeast portion of Louisiana. Next slide, please. And here's some devastation pictures from it. The very top picture is a healthy stand of roseau cane, and this is actually roseau cane is very, very beneficial to storm protection, and it's one of the most erosion resistant species that we have on the coast. And if you look at the bottom left picture, where some stands are being eradicated by the scale, you can see that this may affect some oil and gas drilling sites. And then the second picture at the bottom, that's where a whole stand has been destroyed by the scale. And the third picture is, this also presents a problem because in places where they have been destroyed, we get invasive species like the water hyacinth coming in and taking over. Next slide, please. And here are some pictures of the bugs, and as you can see, they burrow themselves under the first few layers of the plant. If you look at the top right picture, we actually have a native scale, and that's on the right side. And they have very, very low densities compared to these other things because they don't have the natural biological controls in place. And these things can spread for miles in a couple of months. They'll have hundreds on one stem versus where you may have one of the native species on a stem. Next slide. So the important part to understand in this is that these studies take a long time. So as this is destroying the coast, our scientists tell the public that we need 2 years to study them, and the public does not like that because they want to know why we need 2 years to study it. But we need some sort of reference to see how long, how much destruction takes place over how much time. We do have some controls for these, but these are usually controls that are used for small scale house plants where they apply soaps or oils directly to the plants. And the current research is that the current insecticides that they're using are not very effective against the scale. They are thinking about introducing a parasitic wasp. However, these are also from Asia. So whenever we bring another invasive species in from to kill an invasive species, sometimes these other invasive species can have negative effects on the environment. And the newest notes is that the last winter may have helped somewhat, but they're still waiting on data because these things may have killed some of these scales. And then we also concerned because a European variety may be taking over, which is going to be good for the coast as far as protection, but it may be bad for things that do not develop the same relationships, maybe birds that are nesting in European varieties or that won't nest in European varieties. And that is it. Any questions?
Do we have any questions for Damon? Okay. Well, we'd like to thank everybody, all 8 of our graduate students, for giving presentations over these last 2 lecture periods. And remind you that we will be posting the exam later today, and then you'll have until midday on Monday to complete it. Yeah, I think that's all that we need to cover. Mert, is there anything you need to add?
Nope, I think I'm good.
All right. Well, thank you all for a good semester, and we look forward to seeing your exams on Monday.
Good luck, everyone.
Is there a question I hear?
Yeah, I have a question for you guys. If we weren't able to make it on the field trip due to a medical reason, what do we do for the exam portion of that?
We will have to talk to you offline for that.
Okay.